Hello everyone. Today we're going to have the last lecture for this semester. Okay, it's the digestive system physiology part, and please read your book from page 876 to 917 first, and have an idea about what we're going to talk about, and then print out your lecture notes on Canvas and follow the PowerPoint lecture notes for this part of lecture. Okay, so let me get to my slides first. Okay, so we're going to focus on the physiology part of a digestive system, which is the digestion and absorption, the processes for digestion and absorptions. Before we start that, first part, we're going to cover the accessory organs for this system. If you remember, accessory organs are, if you can name them with me, okay, the teeth, tongue, salivary glands, pancreas, and liver. Okay, those are the major accessory organs. Let's start with the tongue first. Okay, so the tongue structure, we actually used this picture before. If you remember, in the immune system, on the base of the tongue, we say we have lingual tonsils right here at the base of the tongue. Okay, so if you look at the tongue structure on the surface, there are these ridges. Okay, first, let's look at these big ones. All right, towards the base of the tongue, there are these V shape lining structures. Okay, if you enlarge each one, it looks like this. This one is called a circumvallate papillae. Okay, so in the circumvallate papillae, there are bitter taste receptors in there. So this is why, if you're taking a pill, if you put on the anterior part of the tongue and then you try to swallow it, and then you don't really feel, taste the bitterness. But if it gets stuck in the back of the tongue, and then you can really taste the bitterness, because most of the bitterness receptors are in the back, in the close to the base of the tongue. Okay, they are located in the structure called circumvallate papillae. Okay, now on the anterior part of the tongue, 2/3 right here, there are two types of ridges. One is called fungiform, another one is called a filiform. Okay, so if you just look at the filiform, let's see, is kind of like this. If you enlarge the surface, it's just rough surface. Okay, surface kind of become like protruded in areas to make it rough. This part of the structure, the main function is try to help to break down big particles. Rougher surface, it can help to break down bigger particles much better than a smooth surface. Right? Another kind of ridges is called fungiform. If it's a fungiform, it looks like this. It's a little bit bigger than a filiform. In the fungiform ridges, there are receptors, taste receptors for, like say, temperature, for sweet, salty, and sour. Okay, so three different ridges on the surface of the tongue: fungiform, circumvallate, and filiform. All right. And then if you have a sagittal view, right in the mid-sagittal side, there is a connective tissue separate the tongue on the inferior side from left to the right, and that structure is called the lingual frenulum. Okay, all right. All right, so in terms of salivary glands, there are three pairs of salivary glands: parotid gland, submandibular gland, and sublingual gland. Parotid gland is the biggest one on each side, and then there's a major duct carry the secretions into the mouth on the upper vestibular part. Okay, so this one, if you maybe you can do an experience. If you just sit here, think someone is chewing a piece of lemon, and then you can feel that you're going to have a watery mouth, and try to feel where these watery secretions came from. A lot will be from this one. Okay, parotid gland. Okay, parotid gland mostly secrete watery secretion. So this kind of secretion is called a serous secretion. Okay, serous secretion. And then submandibular gland, which is labeled right here, submandibular gland. This gland has two major ducts, one on each side, open at the side of lingual frenulum. Do you remember this one I was talking about? This separation on the inferior side of the tongue to separate the tongue from the left to the right side. Okay, on each side of this separation, there is a opening of this duct from submandibular gland. Okay, so maybe you have this experience. You probably saw someone, when they roll up the tongue, they can actually shoot out saliva. That is coming from this submandibular gland. Okay, this gland is kind of we call it a mixed gland. This in terms of a secretion, they have watery secretions. Also, they have mucus-rich secretions. Okay, the third one is called sublingual, which is under the tongue. Okay, sublingual gland. This one mostly have mucus secretion. Mucus secretion. Okay, and for this gland, instead of having one major duct, it has multiple smaller ones under the tongue. Okay, multiple smaller ducts release secretions into the mouth. So the next few slides are the histology slides, okay, about the salivary glands. So on this side is a serous gland. If it's a serous gland, remember I said it's watery fluid, okay, mostly water, some minerals, a little bit of enzymes. So in this case, this gland tissue can stain very well with a dye. So you can see darker stained. Maybe next one I have a better slide, but at least you can see it's a darker stained structure. But on this side is called a mucus gland. The mucus gland secrete mucus. Mucus has lipid component on it, so it does not take the dye very well. So if you look at these mucus secreting cells, they're more like fat cells. Okay, so kind of have transparent looking. All right, so in this slide, you can see these part right here and right here. They're mostly serous secretion gland, but in the middle right here, and here, they're fat-looking cells. Those are mucus secreting cells. Okay, so remember I said mandibular gland is a mixed gland. It has both the mucus secreting and the serous secreting cells. So that's why in this slide you can see both. Okay. All right, this is another submandibular gland, but it's kind of not enlarged enough to see details. But this one is, okay. So this is still submandibular gland, all right? So in the middle right here, fat-looking cells, those are mucus glands, okay? Around it, these darker stained part are the serous secreting part. Let me see if I have an animation. Annotate pen right here. So if I show you right here, okay, this is a typical duct cross section, okay? So you can see cells with a nucleus in there, kind of, all right? So that's the duct structure. And also, if you look at this mucus secreting cell right here in the middle, if you look on the side, you can see a structure kind of like look like a half circle right here. Nucleus. So probably it's like two cells. You can't see the cell membrane over there. So that's a series of secreting cells. So let's try to find another good one, like here. Oh, I did not draw very well. Okay, maybe here. Okay, so basically it gave you an idea. If it's a mixed gland, sometimes you can see structure, histology structure looks like this. The mucus secreting cells in the middle and the serous secreting cells around it in this half circle structure. This half circle structure has a name. It's called a demi-lume structure. Demi means half. Lume is like a moon, okay, half moon structure. So that is a typical mixed salivary gland structure, okay? Serous secreting cells and, serous secreting cells outside and mucus secreting cells inside. Get rid of this. Why it stops? Here we go. Okay, so salivary glands, in terms of components in there, okay, we say water, minerals, and it's a little bit acidic, and the important enzyme is this one. Oops. It is called salivary amylase, okay? Please write it down on the side, okay? I'm going to give you all major enzymes which you need to remember the names and function. Salivary amylase is the first one. This enzyme is used to break down carbohydrate, okay? So it's an enzyme to break down or to digest the carbohydrates, okay? And also there are little bit lysosomal enzymes, which is to take care of minor infections if there's any foreign antigens. And there's a little teeny bit of lipase, which is the enzyme for breaking down fat. So we can just ignore that because it's not a significant amount of that enzyme in the salivary gland. So for enzyme, if we're talking about an enzyme, this is the one I want you to remember. It is called salivary amylase, which can break down carbohydrates. So another way to ask you uh, the question is, if I ask you, when you start chewing a food, okay, what kind of materials can be chemically digested? Choices: carbohydrates, proteins, fat, nucleic acids, or all of them. So the correct answer should be carbohydrates, because in the mouth you have salivary amylase, which can chemically digest carbs. For lipase, remember I said for fat that enzyme amount is insignificant, so we can just say no fat chemical digestion in the mouth. So when you start chewing the bread, okay, when you start chewing the bread, at the beginning you don't taste the sweetness, but if you chew long enough, you can start to taste the sweetness from this food particle. The reason for that is this amylase start to work on the starch and to break down the polysaccharide, okay, and to release small sugar molecules. The small sugar molecules have the sweet taste. How can I get rid of this thing here, right here? Yay. Okay. So the next part is the accessory organs will be the teeth, right? For this part, I'm going to ask you to study this part by yourself. Any structures listed on your lab handout, you should know. It's very straightforward. So you need to know the name of different types of teeth, and you need to know the difference between adult and child's teeth, and also the typical tooth structure from superficial to deep. You need to know the structure. Come on. Stop it again. I don't know. Get it stopped. Okay. All right. Next accessory organ is the pancreas. I kind of went over the structures last time in terms of how the pancreatic juice being released to the small intestine. First, this releasing point to the small intestine, this part is a duodenum. Okay, the duodenum is not very long. It's like a 10 finger lined up. That's the length about the duodenum. And then once the duodenum start to make a very sharp turn, like almost like a 90-degree turn right here, this is the starting point of a jejunum, the second part of small intestine. And in terms of a duct system from the pancreas, all right, this is the major duct, is called the main pancreatic duct, and also have the accessory pancreatic duct. The main pancreatic duct has sphincters which can control how much pancreatic juice being released, but the accessory pancreatic duct is relatively small duct and will continue to release pancreatic juice. And then on this side, the green part is the bile. Okay, bile is made in the liver and then collected by the left and right hepatic ducts and merged to become common hepatic duct, and then continue to travel to duodenum. Once it passes this cystic duct, it becomes the bile duct. Okay, bile duct. All right, and if the bile is too much and it cannot be used all of them, and then the bile will backflow and through the cystic duct and get to the gallbladder and the store over there. So if you look at a pancreas structure, pancreas is considered as both endocrine structure and also exocrine structure. For endocrine part, we already talked about it can make glucagon and insulin, the hormones. For exocrine part function, it can make a lot of digestive enzymes and also bicarbonate ions. Okay, so if you look at this structure on the top here, this drawing, and these are the typical exocrine gland structure. It is a gland specialized from epithelial cells. Okay, so we call them SNR cells. Do I have it labeled here? Maybe it's on the next slide. Oh, right here. SNI or SNR cells. Okay, so these cells will make enzymes and bicarbonate ions, and then these secretions will, or these enzymes and the ions can be transported through the duct system and to the small intestine. Okay, so these are exocrine tissue. And on the side, you have this kind of structure right here, right here. Those are called pancreatic islet. Okay, pancreatic islet. Those are endocrine tissue. So you already know that, right? So these kind of structure, they make hormones. So on this histology picture here, the majority part on this picture will be SNR cells to make enzymes and bicarbonate ions. And this islet structure right here, that is the endocrine tissue. Okay, so this is enlarged exocrine structure on pancreas. So this is the gland. It's made by SNR cells. The cells make enzymes and bicarbonate ions, and then these enzymes will travel through the duct system to the small intestine. Okay, so in terms of what enzymes can be made in the pancreas, here's the list. Proteases, that's the enzymes for protein digestion. Amylase for carb digestion. So think about this. Amylase can be made in the pancreas, but also made in the salivary gland. Okay, lipase is for fat digestion. Nucleases is for nucleic acids. Okay, so like RNA or damaged DNA, any nucleic acids you don't need, and it can be chemically digested using the enzymes. Okay, in terms of the ions, remember I said the bicarbonate ions are the major ones. Okay, so if you remember from last unit, bicarbonate ions can be used to bind with hydrogen ion to adjust blood pH. Okay, so bicarbonate ions can be used to adjust the pH to chelate hydrogen ions, if you can say that. Okay, the duct system is used to transport enzymes and bicarbonate ions into the small intestine. All right, this is another histology picture for pancreas. So this is the islets. These are SNR cells for enzymes and bicarbonate ions. Okay, so now we're talking about the specific enzymes in the pancreatic juice. Remember we talked about there are proteases. Okay, so proteases are the enzymes which can chemically digest proteins. Okay, so let's start from this one. Okay, one enzyme is called trypsinogen. Maybe I'll use my whiteboard for this part. Give me 1 second. Let me get my whiteboard ready. Okay, so we're talking about pancreatic juice, the enzymes. One of them is called trypsinogen. Okay, this trypsinogen, whenever you see this and nogen at the end of the word, that means it's an inactive protein. Inactive protein. So this one can be activated to make a trypsin. All right. So when you have a trypsin, this is the active form of a protease. Okay. Protease. Okay. So this one can digest proteins. All right. So this part of activation need to have an enzyme. It's called well, I let me give you this enzyme later. But this enzyme needs to convert inactive form of the trypsin to the active form of trypsin. This enzyme is made in the small intestine. So basically, this enzyme, when it's firstly made by when it's first made in the right here, when it's this trypsinogen, when it's first made in the pancreas, it is inactive form of enzyme. And then this enzyme is going to travel through this duct system. Once it gets to the small intestine, right here, once it gets to the small intestine, it can be activated, the chemical reaction, you have the enzyme in the small intestine, which can activate the trypsin, and then the enzyme becomes active to used for protein digestion. So the question is, why do you need to have the inactive form of enzymes made first and then being activated in the small intestine? Can you just make the active form and then directly have the active form of these enzymes travel to the small intestine and work there? Well, think about along this duct, these lining cells, they're cuboidal cells, right? So proteins are the major component for cell membrane. If you have proteases, active proteases traveling in this duct system, it can actually destroy your own lining cells. That's not what you want, right? You only want to use these enzymes in the small intestine to digest the food, the proteins coming in from the food. That's why it is advantage to make the inactive proteases first and then activate them in the small intestine. Okay, so besides of trypsinogen, there's another major protease is called a procarboxypeptidase. Okay, Pro is inactive form as well. So this enzyme can be activated to carboxypeptidase and then used to digest the proteins. Okay, so that's another enzyme here. Besides of the proteases, there are amylase. We talked about that's for carbs, lipase for lipids, and nucleases for nucleic acids. All right. So we talk about the pancreas can make digestive enzymes and bicarbonate ions. On top of that, it also can make hormones. Actually, hold on. The hormones, the insulin and the glucagon, which is the endocrine secretions. Okay, so these exocrine secretions can be regulated, definitely can be regulated. Okay, the two major hormones which can regulate pancreatic secretion, exocrine secretion, the two major hormones. One is called cholecystokinin or CCK. Another one is called a secretin. These hormones are made in the small intestine, and then they travel through the blood circulation and get to pancreas. They target on pancreas to regulate pancreatic secretion. Okay, so I want you to remember secretin. In terms of function, there are some overlapping functions, but I want you to remember the specific one, like this one. The secretin is actually more sensitive to pH changes in the stomach. So, for example, if the blood, if the gastric juice is very acidic, pH is very, very low, that is the signal to trigger the secretion of a secretin, which is the hormone, which will stimulate the pancreas to release more bicarbonate ions. All right, so these bicarbonate ions will be released into the small intestine and to neutralize the pH. All right, let me just say it one more time. Okay, so if gastric juice becomes more acidic, that's the signal to trigger the production of secretin in the small intestine. This is a hormone. This hormone will target on the pancreas to stimulate the bicarbonate ion release from pancreas. So bicarbonate ions will be released into the small intestine to neutralize pH. Okay, that's one major function of secretin. Let me give you an example here. Let's say if you just had a Coke, Diet Coke, all right? If you just had a Diet Coke, then you know the Diet Coke has very, very low pH, pH 4, okay? So this juice come into the stomach, so make your gastric juice more acidic, okay? But you didn't really eat anything. You did not eat protein, you did not have fat, you did not have carbs. You know, Diet Coke, there's no sugar in there either. So in this case, there's no use to release a lot of digestive enzymes from pancreas, nothing to be chemically digested, right? Rather, because the pH change in the gastric juice, you really want to have more bicarbonate ions to neutralize this pH. Okay, so in this case, the secretin is the major hormone which will stimulate the pancreas to release more bicarbonate ions to neutralize the pH. Okay, so the next one is the liver, accessory organ, the liver. Okay, so this liver structure, I want you to read your book about the liver structure and then listen to this lecture, and afterwards please review the liver structure. Okay, liver is made by four lobes. Okay, you do need to know the four lobes. Make sure you can identify the lobes. And also this one, falciform ligament. Do you remember this falciform ligament? That is the peritoneum covering the liver and then joined together as a mesentery to connect it to the anterior wall. Right? If you forget about that, go back and review from the previous lecture. Okay, and then next one is called the ligamentum teres. I hope you still remember this from when we talk about the fetal circulation. One difference between fetal and adult circulation is the fetal circulation. There is one umbilical vein. Umbilical vein carries the oxygen-rich blood from the placenta to the fetal side. This umbilical vein in adult, okay, it becomes the ligamentum teres. It is also called the round ligament. Right? Some book will call it the round ligament. Okay. And then the liver, a part of structure is called a gallbladder, which is the location to store the bile. And if you look the microscopic structure of the liver, each lobe is made by hundreds of this structural unit. It's a lobule structure. Okay, so each lobule you see is like a six-sided structure. This one, so the lobular structure are the same for all lobules, and each one is made by this hexagonal structure. Okay, have 6 pointed side. All right, and if you enlarge from one corner right here, one corner, it looks like this. Maybe I have an enlarged one down there. Okay, here. All right, so in the center, there's a major vessel. Okay, at each corner there are at least three tubular structure. It is called a portal triad. Because it has a three tubular structure. Sometimes there are four. Okay, so basically it has a very small vein, very small artery, and then bile duct over here. That's the three triad. And sometimes you have a lymphatic vessel as well. So that would make it four, right? So at least the three for sure. And then in the middle here, this big vessel is called a central vein. All right. So now think about, look at this word here, portal vein. Do you remember where the portal vein came from? From abdominal organs, right? From mostly small intestine and splenic vein, right? So in the portal vein, remember we say it's collected blood from small intestine. So this blood is oxygen poor, but nutrient rich blood. If you remember that from the cardiac system, we talked about that hepatic portal system. Okay, so hepatic portal vein carries oxygen poor but nutrient rich blood. So this blood from portal vein, and then it's going to branch out to form smaller portal venules right here. It's just the smaller or smallest vein right here at each corner, each pointed corner. And here, this portal venule, this blood is going to travel right here, enlarged here, through the capillaries toward to the central vein. On the way to travel toward to the central vein is going to unload these nutrients to the hepatic cells. So this portal blood, it has a lot of nutrients absorbed from digestive system. So once it gets to the liver, okay, it's going to travel from this portal venule toward to the central vein and release the nutrients to the liver cells. And then the liver cells can use these nutrients to make bigger molecules, proteins, antibodies, not antibodies, proteins, albumins. That's a part. That's a example of protein, globulin protein, and also lipid. Okay, so these capillaries are called sinusoid capillaries. If you remember that, sinusoid, they have a much bigger pinched holes and allow bigger molecules to get in and out, right? So nutrients will be released to the hepatic cells. At the same time, the bigger molecules made by hepatic cells can get into the sinusoid and then get to the central vein. Okay, so, but this portal vein, it carries the oxygen poor blood. So all these hepatic cells, they survive, they need to have oxygen. Where the oxygen came from? It's from systemic circulation. If you remember, the common hepatic artery branched out from celiac trunk. Remember that? Common hepatic artery. And then branch out to form hepatic arteries toward to the liver. And at each corner right here, from the hepatic artery to branch out, form these portal arterioles. And these arterioles carry oxygen-rich blood. This oxygen-rich blood is going to travel through the sinusoid and going toward to the central vein as well. Okay, this way is going to unload oxygens for the hepatic cells. All right, so far I gave you two paths. One is the portal vein, okay, from portal venule, the nutrient-rich blood traveling toward to the central vein. And another one is from portal arteriole, is oxygen-rich blood travel through this capillary bed. So both of these are blood traveling paths. One is for nutrient exchange, another one is for gas exchange. Okay, so remember at each corner it's called a portal triad. So I gave you two already. One is portal venule, one is portal arterial. The third one is called right here. Where is that? Do we have it labeled here? Bioduct. See this? Bioduct. Okay, so what is the function for bile duct? Is to collect the bile made by hepatic cells. So all these liver cells, they can make bile, and the bile will be collected into the small tubular structure is called a canaliculus, bile canaliculus. This word, you learn this in 430, the smaller, very small tubular connections between the bone cells are called canaliculus as well, right? So all these canaliculi collecting bile and then eventually merge into the bile duct right here, and then will be collected to, let me go back to this, let me review this one more, oh, right here. Okay, so and then the bile duct will merge into the left and right hepatic duct, and then common hepatic duct, and then bile duct, and then to the duodenum. Okay, that is the path. All right, so one more time, on this diagram, you see the blood is traveling toward to the central vein. Okay, blood is either coming out from portal vein or portal arterial. They both travel toward to the central vein, and the bile is traveling in the opposite direction. Is going to be collected in the bile duct. Okay, and then travel this way and go toward to the duodenum. All right, so this is a histology picture for liver. Okay, at each portal triad, you should be able to see three tubular structure. Okay, so arteries and artery and vein, arterial and venial. So remember if you compare artery to the vein always have a bigger lumen, right? Arteries have more muscular structure, so that's what you can see over here. This is the arterial side, this is the venial side, and then here is more like a cuboidal lining duct structure, lumen inside. Okay, this is the bile duct. Okay, so if you see a histology here, mostly hepatic cells around, and then you can see this portal triad structure, have an arterial, have a venial, have a duct. That is the liver histology picture. Okay, so let me skip this one. All right, so bile release. All right, so here we go. Do you remember this one? CCK is a hormone made from small intestine. All right, it can target on pancreas. It also can target on bile release. All right, so I say there's some overlapping functions for CCK and secretin, but they do have more unique functions. For secretin, it can stimulate pancreas to release more bicarbonate ions. But for CCK, it can help to release more bile. Okay, so now think about the function. If you just had a big steak, okay, a lot of fat, right? So you need to digest the fat. Then you really want to release more bile to help digesting the fat particles. And then in this case, you're going to the CCK making cells will be sensed and start to making more CCK, and then CCK will target on bile release to release more bile into the small intestine. Okay, so here is CCK can do the following to release more bile. Alrighty, so now we're done with the accessory organs, and then we can actually focus on the chemical digestions. Maybe I should stop here I said this is the last lecture for this unit. Maybe let's break it down into two lectures so I can stop here. So this one is only covering the accessory organs, and then I will have another lecture focus on the chemical digestion and absorption. I think that's better. So let me stop here. So this is the lecture number 2. Okay, you're going to have a lecture number 3 on digestion and absorption coming up.